During pregnancy, the fetus depends on the mother for nutrients and gas exchange. Because the infant cannot breathe while confined, its blood has a unique circulation path to ensure adequate delivery of oxygen and nutrients to the fetus. Remember, the lungs of the fetus are not participating in gas exchange, so blood must bypass the pulmonary system to provide oxygenated blood to the body. If the blood is unable to shunt past the pulmonary system, oxygenated blood will not circulate through its body. Fetal blood flow begins with the mother after the placenta is implanted into the uterus. Maternal blood enters the basal plate through the spiral arteries, then perfuse into the intervillous spaces and flows around the villi where oxygen and nutrients are transferred into fetal blood. After maternal blood passes through the intervillous space, it drains back into the basal plate and returns to maternal systemic circulation through the uterine veins. The villous capillaries bring fetal blood close to, but not in contact with, maternal blood. From there the blood travels to the umbilical cord. It is important to note now that this process is complex and numerous factors, such as maternal health and diseases, can affect how well this part of the system works and will be discussed in detail later. Fetal circulation relies on 3 shunts. First is ductus arteriosus, which is a hole between the pulmonary artery and the aorta. It serves to divert blood away from the fluid-filled lungs towards the descending aorta and placenta. Now take a closer look at the location of this shunt in relation to the 3 branches of the aorta. The first branch is the brachiocephalic trunk, which branches into the right subclavian artery and supplies blood to the right arm, and the right common carotid artery that supplies blood to the head. In the middle is the left common carotid artery. And the third is the left common subclavian artery. Then blood travels through the descending aorta to the rest of the body. Look how close the duct is to the second and third branch, while the first branch is furthest away and heads to the right arm. Blood samples or pulse oximetry readings taken from the right arm are considered preductal. The second and third branches may mix with blood shunted through the duct from the left atrium. Any blood samples taken from the low extremities is considered postductal. Comparing differences in measurements from preductal blood and postductal blood can provide hints that a congenital heart defect may be present. Next is the foramen ovale. This is a small opening between the right and left atrium that allows blood to cross the atrium and bypass pulmonary circulation during fetal development. From there, the blood goes to the left side of the fetus's heart and provides oxygen to the fetus's brain. Last is the ductus venosus, which is a fetal vessel connecting the abdominal umbilical vein to the left portion of the inferior vena cava just below the diaphragm. The function of the ductus venosus is to shunt the substrate-rich blood coming from the placenta via the umbilical vein to the heart. Now let's follow blood through fetal circulation. Fetal blood flow begins at the placenta where it picks up oxygen and nutrients from maternal blood. From there, it moves through the umbilical vein towards the infant's liver. Blood is then shunted through the ductus arteriosus. Some of the blood travels to the liver while the rest descends the inferior vena cava into the right atrium. From the left atrium, blood is pumped into the left ventricle, then out to the body through the ascending aorta and is sent to the rest of the body. Blood returns from the body into the vena cava where it enters the right atria and mixes with the blood coming from the placenta. The blood is pumped into the right ventricle and is pumped into the pulmonary arteries where some of it goes to provide nutrients to the growing lungs while most of the blood shunts through the ductus arteriosus into the descending aorta. The descending aorta connects to the 2 umbilical arteries and travels to the placenta where waste products and carbon dioxide are released into the mother's circulation and oxygen and nutrients are picked up and travel back to the fetus.
Nah, they're not ready for that yet, dude.
When fetal and maternal blood come in close contact, gas exchange occurs. This process can be further explained by the oxyhemoglobin dissociation curve, which describes the relationship between available oxygen and the amount of oxygen carried by hemoglobin. Each hemoglobin molecule can bind 4 oxygen molecules, but each hemoglobin molecule is not always fully saturated with oxygen molecules. If the hemoglobin is completely full with 4 oxygen molecules, its oxygen saturation would be 100%. If the hemoglobin has no oxygen molecules, it is called deoxyhemoglobin and its oxygen saturation would be 0%. The P50 describes when hemoglobin's oxygen saturation is at 50%. When the P50 moves to the left or right, it is called a left or right shift. A right shift would make the P50 higher on the partial pressure of oxygen axis. A left shift would make the P50 lower on the partial pressure of oxygen axis. Numerous diseases and processes can move the cure left or right. A right shift indicates there is a higher partial pressure of oxygen in the blood, since there is essentially extra oxygen or the tissues are requiring more oxygen. Hemoglobin will be more likely to unload oxygen from hemoglobin. The hemoglobin would not want more oxygen, so it would decrease its affinity for oxygen. A left shift indicates there is a lower partial pressure of oxygen in the blood. Since there is less oxygen, the hemoglobin try to keep the oxygen it has, or prevent oxygen unloading, or try to pick up more by increasing its affinity to oxygen. Carbon dioxide and pH will move the curve in different directions. If there is a high concentration of carbon dioxide, such as during increased metabolic activity, this can decrease the pH, making the hemoglobin unload oxygen to facilitate the metabolic demands of the tissue. This is a right shift. The increase in metabolic activity would also raise the temperature, making hemoglobin want to unload oxygen to the tissues that are requiring more it. Another right shift. If there is a low concentration of carbon dioxide, it could indicate that there is a decrease in metabolic activity, which would increase the pH, making the hemoglobin want to keep its oxygen molecules. This is a left shift. The decrease in metabolic activity would come with a decrease in temperature, making the hemoglobin want to hold on to its oxygen since the tissues are not demanding it. Another left shift. 2,3-DPG is a byproduct of glycolysis and controls how much oxygen is released from hemoglobin. When the body lacks oxygen, it can signal an increase in the output of 2,3-DPG. This would mean the tissue needs oxygen and the hemoglobin should unload it or decrease its affinity. This is a right shift. When the body has decreased tissue metabolism, it signals a decrease in 2,3-DPG to encourage the hemoglobin to pick up more oxygen or increase its affinity for oxygen. This is a left shift. Lastly, fetal hemoglobin is a special type of hemoglobin that has an extremely high affinity for oxygen, making it want to pick up oxygen molecules, causing a left shift. To review, a left shift increases hemoglobin's affinity for oxygen. This can be caused by an increased pH or becoming more alkalotic, decreasing carbon dioxide, decreased 2,3-DPG, and fetal hemoglobin. A right shift decreases hemoglobin's affinity for oxygen. This can be caused by a decrease in pH or becoming more acidotic, increasing carbon dioxide, or increasing 2,3-DPG. Pregnancy can also move the curve right. The oxygen carrying capacity of blood depends primarily on the hemoglobin molecule. Fetal hemoglobin is a principal oxygen carrier in both preterm and term neonates. Blood from the placenta to the fetus has an oxygen saturation of about 80%. The low oxygen saturation of the fetal circulation is a significant and important characteristic. The fetus's hemoglobin is unique because it contains fetal hemoglobin. Compared to adult hemoglobin, fetal hemoglobin exhibits a higher affinity for oxygen. The movement of oxygen and carbon dioxide between the mother and fetus are facilitated by their concentration gradients, where molecules will travel from areas of high concentration to areas of low concentration. This process is called diffusion. Since the mother's hemoglobin concentration of oxygen is high and concentration of carbon dioxide is low, and the fetal hemoglobin has a higher affinity for oxygen with low content, and higher amounts of carbon dioxide. When their blood comes in close contact in the placenta, oxygen molecules will diffuse from the mother to the fetus while the fetus diffuses carbon dioxide molecules to the mother. Another aspect for the maternal and fetal transfer of gases is explained by the Bohr effect, which states that hemoglobin's oxygen-binding affinity is inversely related both to acidity and to the concentration of carbon dioxide. In terms of fetal circulation, the Bohr effect occurs twice in the placenta, once on the fetal side and once on the maternal side. This is called the double Bohr effect. On the mother's side, as more carbon dioxide is diffused from the fetus, it makes the blood slightly more acidic. This will shift the curve towards the right, which causes more oxygen to be released from the hemoglobin. On the fetus's side, as the carbon dioxide concentration decreases, it makes the blood slightly more alkaline, shifting the curve left, facilitating oxygen uptake by the hemoglobin. To review, 3 factors facilitate the gas exchange between the mother and fetus. First, the concentrations differences in oxygen and carbon dioxide facilitate diffusion. Second, fetal hemoglobin has a higher affinity for oxygen compared to adult hemoglobin, and the hemoglobin of pregnant women have a decreased affinity for oxygen. Third, the double Bohr effect.
Nah, they're not ready for that yet, dude.
Amniotic fluid is the yellowish liquid that surrounds the fetus during development and plays numerous important roles in fetal growth. First, amniotic fluid provides the fetus with room to grow. It also serves as a cushion between the fetus and the umbilical cord, as well as between the fetus and the uterine wall. Amniotic fluid also helps protect the fetus from infection because it has antimicrobial properties. It also serves as a reservoir of fluid and nutrients for the fetus, because it contains proteins, electrolytes, immunoglobulins, and vitamins from the mother. Amniotic fluid is 98% water and electrolytes. The other 2% are signaling molecules, peptides, carbohydrates, lipids, and hormones. During the early stages of gestation, the water in amniotic fluid is derived mostly from maternal fluid. At about 10 weeks, the fetus begins to produce urine, which is secreted into the amniotic sac. In the 2nd and 3rd trimester, fetal urine becomes the largest source of amniotic fluid, along with smaller amounts of fetal lung fluid, gastrointestinal secretions, and excretions from the umbilical cord and placenta surface. Proper amounts of amniotic fluid require a balance between production and consumption. The fetus requires a specific amount of amniotic fluid for proper development. Numerous factors can create situations where there is too little or too much amniotic fluid. Either extreme can indicate problems which are discussed in detail later. Respirations, swallowing, and urination are the main routes of exchange between the fetus and amniotic fluid to maintain the fluid balance. Fetal reabsorption of amniotic fluid occurs primarily by fetal swallowing of amniotic fluid and by intramembranous absorption to a lesser degree. Fetal swallowing represents the major route of amniotic fluid reabsorption during intrauterine life and plays an important role in gastrointestinal development. If the infant is unable to swallow amniotic fluid, the fluid will build up, which is called polyhydramnios, and can be caused by mechanical or neurological barriers to swallowing, overproduction, or decreased absorption. If the infant is unable to produce adequate urine, there will not be enough amniotic fluid and is called oligohydramnios. This can be caused by conditions where amniotic fluid is lost, underproduced, or idiopathic.
Nah, they're not ready for that yet, dude.
While the fetus is confined during pregnancy, the lungs are growing and are filled with fetal lung fluid. This fluid is different than amniotic fluid. It is produced by the epithelial lining of the developing lung. As it is produced, it fills the lung by maintaining them in a distended state. This distension of the alveoli stimulates the lungs to grow and develop their vasculature and, eventually, secrete surfactant. The fetal lung fluid contains phospholipids, plasma, and lymph and has a high concentration of chloride due to the increased secretion of chloride from the tracheal and distal pulmonary epithelium. The fetal lung is continually producing fetal lung fluid at a rate of 4 to 5 milliliters per kilogram per hour. Extra fetal lung fluid leaves the lung via the trachea and is either swallowed by the fetus or enters the amniotic fluid. The fetal lung fluid that combines with amniotic fluid can be sampled through amniocentesis and provide information about the degree of lung maturity. Later in gestation, fetal lung fluid alone makes up as much as 1/3 of total amniotic fluid. One of the tests of fetal lung maturity is the lecithin to sphingomyelin ratio or LS ratio, and the levels of phosphatidylglycerol or PG. With the LS ratio, lecithin is produced by the fetal lung while sphingomyelin is a non-fetal membrane lipid found in amniotic fluid which begins to decrease by about 32 weeks gestation. The higher the ratio of lecithin to sphingomyelin starting at 2 to 1 indicate lung maturity. The presence of PG increases in production by about 35 weeks gestation. If it is present in certain quantities, this indicates lung maturity. Fetal breathing movements are breathing-like movements that occur episodically in healthy fetuses during much of gestation and can be seen as early as 11 weeks gestation. This breathing movement requires organized and rhythmic contractions of the diaphragm, as well as some skeletal muscles related to breathing, like those in the chest wall and upper respiratory tract and the neural regulation of breathing. These breaths play no role in fetal gas exchange since the lungs are completely filled with fetal lung fluid. However, the breathing movements play an important role in lung growth and development. When the fetus breathes, fetal lung fluid moves up the trachea where it is swallowed or leaks into amniotic fluid. The pressure in the fetal trachea is higher than the pressure of amniotic fluid, and this maintains a distending pressure in the lungs which stimulate lung growth. Fetal breathing also increases cardiac output and blood flow from the heart to the placenta. Any fetus with disruptions in the normal development and progression of fetal lung fluid or fetal breathing movements can experience inappropriate or maldevelopment of the lungs. The lungs undergo specific phases of development during gestation, starting out as lung buds and continue to grow until about the 8th year of life. Surfactant is a complex mixture of phospholipids and proteins that are synthesized and secreted by type II alveolar epithelial cells, also called pneumocytes. The type I alveolar cells are responsible for gas exchange. Surfactant production begins at around 24 weeks gestation, but it is not produced in adequate amounts until about 32 weeks gestation. While gas exchange in the lungs can occur before an adequate amount of surfactant is present, it is extremely limited. A fetus delivered before 32 weeks gestation will lack many of the structural components required for adequate gas exchange. Surfactants are chemicals that reduces surface tension. In the fetus, pulmonary surfactant works to reduce surface tension within the alveoli, allowing them to open, remain distended, and participate in gas exchange. Surface tension is a property of liquid that refers to the cohesive forces between molecules at the surface of the liquid. This allows the liquid surface to act as an elastic membrane when stretched. At liquid-air interfaces, surface tension results from the greater attraction of liquid molecules to each other, rather than to the molecules in the air. The alveoli are the microscopic, balloon-shaped structures located at the end of the respiratory tree. They contain collagen and elastic fibers that allow them to stretch when they are filled with air during inhalation, then spring back during exhalation. However, they remain slightly inflated at the end of expiration. Within the alveoli is liquid that creates surface tension. This surface tension is the result of the greater attraction of liquid molecules to each other, rather than the air. Since the alveoli is a sphere shape, surface tension works to pull or hold the elastic sphere into its smallest state. The sphere will resist opening or inflating unless a high amount of pressure is applied to force it open. When the alveoli are forced open, they can become damaged, presumably because of shear stress on the alveolar walls. The application of surfactant to the elastic sphere reduces the amount of pressure required to expand or inflate the sphere by decreasing surface tension. When surfactant is present in the alveoli in adequate amounts, a film develops that allow the alveoli to expand with minimal pressure and avoiding the damage that developed from using high pressure. After birth, the fetal lung fluid is expelled from the lungs, and from each individual alveoli. With adequate surfactant, the alveoli have a coating of surfactant and are easily opened and participate in gas exchange. When an infant is born without adequate amounts of surfactant, such as when they are premature, the alveoli have a high surface tension and it will require a high pressure to open, which damage the alveoli in the process. This can lead to respiratory distress in the infant. The fetus growing from an egg to an infant is truly amazing. Next, we'll examine how the fetus adapts to extrauterine life.